I think. Communication. I will stop, ask question, but at any point of time you can stop me, you can ask question. So what is a metabolic network? I think we have started the discussion yesterday. We have discussed that a metabolic network is a network of metabolites where the nodes are metabolites and the edges are basically represent reactions, right? So in the first lecture, basically I'll give you a brief overview of a metabolic network model, how we can build it. In the second part of the lecture, I will talk about how can you build a metabolic network model for a photosynthetic microbe. And tomorrow, we will discuss how you can build a model for a cyanobacteria. So you could see that I'm starting from here, the most generic metabolic model building procedure. Then I'm talking about how you can do that for a photosynthetic microbe. And by now, you should know that photosynthetic microbes could be of different types. It could be a cyanobacteria, it could be a purple sulfur or non-sulfur bacteria. But tomorrow, we will talk mostly about cyanobacteria, a specific kind of a photosynthetic microbe. Make sense? All right. So the first step of a model building process is to understand the central dogma of biology. I think in the last tutorial, I also mentioned that when we build a metabolic network model, we basically operate in the reaction domain, right? That's what the metabolic network model is built upon. But what you need to do, you need to start from the genome. You need to know what are the genes in a given system, in a given microbial system. It could be a photosynthetic one, it could be a non-photosynthetic one. Regardless of the type of the microbe, you have to start from the DNA. In other words, you need to know what are the genes that are there. So once you have the genes, basically the genes can produce mRNA, they can produce other sort of RNA as well, but we are interested about mRNA because we would like to get to the protein. So what are the proteins in that given system? And again, not all the proteins will have metabolic functions. Proteins have multiple different other functions, but for the sake of model building, we are interested about those proteins which have a metabolic fingerprints, or in other words, we are interested about proteins which can catalyze the reactions. So long story short, if you move through this direction from DNA, RNA, protein, at the end you will have reaction. You will basically have a list of reactions that can potentially occur in a given system. Now, having known this mapping, what are the issues that you can think about? We all understand that this is the central dogma of biology. One additional thing that you are learning today is from protein to reaction. Usually, when we study the central dogma, we end or we stop in the protein level and that's it. For the sake of building a model, you need to know the reaction or list of reaction. So what do you think the issues could be in this mapping? Can you think about one or a few?
One protein can participate in multiple reactions.
One protein can participate in multiple reactions. In other words, multifunctional protein. Yes. Degenerative protein can be formed. So in other words, a protein can change its function. Anything else? Active site might be blocked. So in other words, modifications in the protein level. Anything else? So what about those genes which have unknown or hypothetical functions? Right? Even in E. coli, as I said, if I remember correctly, 35 to 40% genes do not have any functions, do not have or have hypothetical functions. Now that was in E. coli, the mostly studied organism, and now for the rest you could take a wild guess that what that will be, right? So that will be a huge issue when you are building a metabolic network model. So, for that there are ways you can overcome these challenges, which we will discuss in the next few slides. But from the very beginning, I need you to understand what are the challenges that you need to face. What the things that the other things that we have discussed, that multifunctional protein, activation or inactivation of proteins, these are stuff that the model can handle as long as you have a reaction associated with the protein. So, the way the model can handle it, let us say, to give you a very crude example, in one condition a protein is active, in other condition it is not. How can you take that account in a model? Because in the model you do not have proteins, right? What do you have? Just reaction. What do you have? You all have the flux through the reaction. So, can you guess how you can take that into account? So, the question is protein to reaction. So, you have two proteins, sorry, you have one protein, two different state. So, you have one protein and you have two different state, state 1, state 2. What I am asking, in state 1 it is active, state 2 it is inactive. The question is, how can you take that into account in a metabolic model? Because in metabolic model you do not have any protein. Absolutely. So, no protein, no flux for sure. Even when the protein is active, the flux might be a function of many other things, but this is absolutely sure that when the protein is not active, there will not be any flux. So, again this is a very simplified and crude example. So, let us say protein P catalyze reaction 1. The way you model this is V reaction 1 will have non-zero value, whereas V reaction 1 will be equals to 0 in this case, right. Now, about all these other things, a protein being multifunctional, a protein being a protein complex, these are other issues that you need to consider. You need to think about the logical relationship. In metabolic model domain, these are known as GPR and ORs. So, GPR is basically gene protein reaction association. If you know the reaction, this GPR association, and if you can define the and or relationship in the gene or the protein level, that will help you to determine this kind of constraints in the metabolic flux. Because at the end, everything has to be interpreted based on reaction flux. Are we clear? So, again, just to repeat myself, whatever happen in the DNA, RNA or the protein level, if you are operating in the metabolic model domain, everything, every piece of information has to be interpreted in the form of reaction, or in other words reaction flux. Okay. So, now for today's tutorial, one of the tools that we will use is called KBase. Have all of you registered for KBase? Some, I could see some nods. If you haven't, please try in the break. If you cannot, then we will do it in the tutorial. So, that is one of the database or tools or set of tools people started using in this area. What are the advantages? The advantages are, if you are not an expert user, for instance, some of you work in experimental biology. So, in your day to day life, you do not deal with model, although you want to use the model to inform your experiment. So, the tool will help you to do that. So, it is kind of a black box. You may not want to know every single detail, but as long as you have a basic understanding of metabolic model and how flux balance analysis works, you can do that. So, that is what KBase is. And other than that, some of you, I am sure you heard about KEGG, UniProt, which is a protein database. This one is also a protein database, and there are also biopsych, all this psych which basically contains mostly reaction and protein information of different organisms. So, these are publicly available resources that you might want to use when you are building a metabolic network model. All right. So, now comes the question, as I was telling you, how can you define the GPR relationship? So, GPR again is gene to protein to reaction association. So, let us take these 3 cases as examples. So, what is happening here? We know about this enzyme succinate dehydrogenase. You could say, and again these examples are taken from E. coli, but these are just examples to prove my point, like what do I mean by the complex relationship in the gene or protein level. So, you could see that in that succinate dehydrogenase there are 4 there are 4 genes involved, right? 4 genes. 4 genes can produce 4 different peptides, and with these 4 peptides can be assembled to a protein, and this protein can catalyze 2 reactions. So, it is a very complex relationship, right? You could see that in the gene level or in the protein level it's an AND relationship. You need to have all these genes to produce all these peptides, so that you can have this one protein, and that protein itself is a multifunctional protein, right? It is a multifunctional protein because it can catalyze 2 different reactions. What about this? It is a transporter. It has to produce these 3 different peptides. These 3 peptides could be assembled to 3 different proteins, and these 3 different proteins are basically protein subunits, and these 3 could be assembled to a protein complex which can catalyze 1 reaction. So, even in here you can see an AND relationship, right? You need to have all these genes, all these proteins to have this reaction activated. So, if I tell you so let us discuss about it. So, if I tell you that this gene 721 does not express in a given condition, do you think there will be flux through these reactions? No. Just do an AND and OR relationship. So, whenever you are building a model, you need to define this relationship. If you do not define these relationships, you will not be able to publish that work for sure, because it is of no use. So, let us say I have a microarray or RNA-seq data. If I have the proteomic data, protein abundance data, I would like to incorporate the regulations in the model. If you do not give me this logical relationship, then how can I do that? What is the point of doing this exercise? The other argument is, what about experimental biology? What about synthetic biology? Because when we work in a lab environment, does any of us like look at an organism and try to find out the reaction? No. We focus on the genes or the proteins, right? So, what I am trying to get here is that without the logical relationship, you cannot take the results from the model back to the experimental domain. You see what is happening here? I am basically explaining the information flow. You as a modeler operate in the reaction domain. I as an experimental biologist operate in the gene or the protein domain. So, we need to understand each other. We need to understand how this information flows, and if we understand that, then and only then we can design good experiments. Does it make sense? So, for this case, the third example, again you could see three different genes, and these 3 different genes can produce 3 different peptides. I think the pointer is okay. These 2 different peptides can be assembled to a protein complex, but you could see that these 2 proteins are kind of isozymes. So, if you have just one of them, your reaction will be active and it could carry flux. So, in other words, if I tell you if I deactivate B1779, will the reaction carry flux? Yes. If I say if I deactivate this gene, will it carry flux? So what needs to happen so that the reaction will not carry flux?
Either 1, 3.
So 2 of these 3 genes have to be deactivated, right? So again, you just perform an experiment that I just mentioned. As a modeler, I gave you a problem that I would like to make sure there is no reaction flux to this reaction, and then you interpreted the information to an experimental biologist. You said that okay, you could like take this gene and take one of these 2. If you take them out, if you mutate them, you are good. There will not be any flux. Make sense? Okay. Quickly recap. We have discussed so far how could we establish GPR relationship. The reason why we need to establish GPR relationships so that we can interpret results from the experimental end, and we can also pass the information from the model to an experimental biologist to perform real experiments. That's why we need to have this information. Any confusion, doubts, concerns? All right. So I think this would be one of the slides that we all know about. Like when we learn these different steps in biology, basically we know what is catabolism. The term itself means kind of breaking down. So you have a carbon source, you are taking it in, you are reducing them, basically breaking them into multiple different products, and then we all know what is anabolism. You are assembling them and you are making multiple other products which will be useful for growth, for many other cases. And we also know we need to have an energy metabolism so that we can power or we can support the energy that is required to perform anabolism, because we are assembling things. When you are assembling things, you have to spend energy, right? When you are breaking things down, basically you are breaking things down so that you can produce energy. But when you are assembling something, you have to produce energy. So you have to have an energy metabolism. We also have discussed yesterday that we have to define the macromolecules when we are defining biomass. To give some examples, proteins, RNA, DNA, and like other stuff. Yeah, and cells sometimes also produce some product of importance. What do I mean by that? Why do you think a cell has to produce something and excrete when it's growing? Do you see a reason why it has to do it? Why it doesn't just, so what I'm basically asking, in the exponential growth phase, when we expect the cell to grow, basically in this phase, why do you think sometimes some of the biological systems excrete something? Why? Toxic for the cell, so it will excrete that. Sometimes for defense. Like if it's under stress, it would like to create something, excrete, so that it can get rid of what it's affecting. Anything else? What about the byproduct? If you remember that pipe flow diagram I told you, if you have a growth coupled product. So in other words, what I'm saying, when you are growing, as a byproduct, something has to be produced, because without producing that product, the cell will not be able to grow. When it produces something, it cannot keep everything inside, so it will just excrete. So in other words, it's balancing the carbon flow, or it might have produced so much energy that it has to spend something on. So then it will excrete. So that's what I mean by the important specialty products. Any questions? All right. So far we have discussed what is GPR, why it's important to establish the logical relationship, how we can interpret the results, from the experimental side, and how as a modeler we can pass the information to an experimental biologist. Then we have discussed catabolism, anabolism, and why we have to maintain an energy level, what is a growth coupled byproduct formation. The next is why we need to balance a reactions, why balancing is important in a modeling framework. We all know, like we know that a reaction has to be balanced, but when you are developing a model, let's say hypothetically you have bunch of reactions which are not balanced, what do you think will happen? So what I am asking, in your model which has 1000 reactions, hypothetically you have 50 reactions that are unbalanced, what will be the effect if you operate with unbalanced reactions?
When we are calculating the biomass or doing some other experiments, then we might get erroneous data.
We might get erroneous data. Okay. Anything else?
The flux would be different without the balancing because it has to multiply with the factor.
The flux would be different. In other words, erroneous results. Anything else? People at the back, what do you think? Sorry, again? The desired product may be different. Okay. Anything else? Yes. Yes to all this. But above everything, tell me again. Conservation of mass. You are violating conservation of mass. Yeah, we know that. But let's say we don't want to deliberately have reactions which are not balanced. But hypothetically, let's assume that because these are processes, and whenever you are doing something like this, obviously you will make errors. We are humans. We make errors, right? So I'm saying even after the refinement, you have some reactions which are not balanced. What will happen then? That is the question actually. I know that I am violating the conservation of mass. I know that. The biggest concern is, let's say, let's take the example of a carbon imbalance reactions, right? You on the left side, let's say you have 2 carbon, on the right side you have 5. So you are making carbon out of nowhere. When you do that, down the line, what the metabolites which are connected to other reactions, that will ensure that it's possible you may not have to take that much of carbon source from outside, because you are already generating out of the thin air carbon inside the cell, right? That doesn't make sense. First of all, this kind of metabolic model will give you only qualitative results that you have to take with a grain of salt. I think I have discussed with the examples that I showed yesterday that how you take these examples, these case studies, then you have to verify in the experimental setup. Now that's one thing. So some of the model predictions will not be right for sure. On top of that, if you operate on a model which is not even of better quality, what's the point of doing this modeling exercise? There is no point. You will give like some bad results to an experimental biologist. He or she will do the experiments. Most likely none of them will be right. So you are wasting everyone's time. If you remember, the whole point of using this modeling technique so that we can design better experiment. So this is failing the purpose, right? Are we clear on that? So do we understand why balancing is important? So we need to establish the GPR relationship. We need to make sure the model reactions are balanced. Now here is a comment, and you might think that I am bragging about myself, but even if you take the models from very good groups which have been publishing models for 15 years, you will see so many reactions are not balanced. But if any of you would like to work in this area, you want to absolutely make sure that all the reactions are balanced, because otherwise you are not helping the experimental biologist. All right. So other issue that we face is, is there anyone with electrical engineering background? No, but we have taken electrical circuit design class regardless of our background. If any of you has an engineering background, you have to take it. I know that. I took it. I am a chemical engineer. So, Kirchhoff's loop law, how many of you heard about it? Can any of you just explain what is it? Go ahead. Yeah. Yes. So, in other words, if you are going through a loop of a circuit, electrical circuit, the sum of current will be 0. Similar concept in a metabolic network model. If there is a loop in steady state, the flux through the loop will be 0 unless you have a driving force. Unless you have a driving force, that should be the case. Similar concept. Now, I think I have a slide at the end. Let me show you that. Okay. So, let us focus on the first diagram, A. So, we have a very small network. There are 3 different metabolites. We can convert C from A either through this one step or through these 2 steps. So, A produces B, B produces C. By looking at the arrows, you could easily assume that we are talking about reversible reactions. It could go in the other direction as well. And when you look at the numbers, these are the possibilities. So, the reaction flux could at most be 10, and again I am throwing some numbers. So, the flux through this reaction will be either positive 10 or negative 10 depending on the direction, and likewise for this reaction and for this reaction. Now, let us focus on this. Let us look at this diagram and let me know, what do you think? Do you think it is a problem? And if you do, let me know why. If you think it is not a problem, still let me know why. What are the issues here? Think about it. Anyone? So, these are the values that you will get just by doing flux balance analysis. So, the first values are basically the ranges of the flux, and the second is what you will get. Let us say hypothetically, if you do a flux balance analysis with a biomass as an objective, as we have discussed, and then you will get these flux values. Why do you think it could be an issue, or why do you think it is not an issue? First of all, so let me narrow down the answer here. Do you think the mass balance, mass conservation is violated here? Let us consider the balance for A, B and C. Do you think they are balanced? So, what is the input for A? So, 1 plus 9, and what's the output? 10, right? So the mass conservation is good. What about C and B? What about B? What's the input? And the output? What about C? So the output is 1 and 9, 10, the input is 10. So can we all agree that mass conservation is fine? Do you think still we have a problem here? We are confused. Okay. Let me change this number. Now focus on this board. So I am changing the number. If you have difficulty with my handwriting, it's basically 1000,999,999. Do you think mass balance is good? So again, if this is 999, this is 1, this is 1000. Do you think mass balance is fine for this case? What will happen if I increase this number to 10,000, 9,999 and maybe even more? Do you see the point that I could have a million here and yet the mass balance will not be violated? Do you think that happens in real life? Do you think we can get that much of flux, let's say hypothetically, when we are using 1 glucose? Do you think a biological system works like that? Again, think about it. Do you think it's an issue? First of all, if you don't understand with those small numbers, focus on these big numbers. Replace 1000 with 10,000, a million, a billion. You can have anything. What would be the limiting factor here? The limiting factor would be, let's say, for these fluxes V1, V2 and V3, you have a limit. So let's say the limit is negative 1000 to positive 1000, and same thing for the rest. What will happen in real life? You will always get this maximum value. If it's 1000, you will get 1000. If it's 10,000, you will get 10,000. But in real life, in practical scenario, that doesn't happen. So can we all see this is a problem? Can we all see that this should not happen? So this kind of loop is known as thermodynamically infeasible cycle, because as I said, same as Kirchhoff's law, in steady state through the cycle, the flux, the net flux would be 0, which is not the case in here. The net flux through this is not 0. So when you build a model, two things you need to remember: the reactions are balanced and your model doesn't have any infeasible cycles like this, because if you have it, this is what you might end up with. For some of the reactions, you will have these weird flux ranges which will have 0 biological significance. Are we good? So what is the confusion? Because I think there might be some confusions. Yeah. How do you identify infeasible cycles? So there are many different ways. One of the ways is when we operate. Let me show it on the board. So when we operate in a biological system, this is the boundary, system boundary, right? So the way we do flux balance analysis is based on what's going in, and most of the times what's going in would be your nutrients. So would be glucose. For photosynthetic microbe, it could be light, right? So for the sake of this discussion, let's limit ourselves to glucose, just plain and simple. So you provide glucose and you define an amount, and that depends on the medium, how much glucose you provided in the medium, and then when the cell cycle is done, you measure it, you know how much is uptaken, that would be your amount. And based on that, basically so this is central carbon metabolism basically. You can define the flux ranges. So let me ask you this question. When I say 0 glucose for my model, does any of my reaction carry flux? There is no nutrient. So should there be any flux? There should not be any flux, although there might be some small amount flux through some cycles which already exist, which are not infeasible cycles. For instance, TCA cycle is a cycle which exists, and we know that this is not an infeasible cycle. Urea cycle is a cycle which we know it exists, and it's not a thermodynamically infeasible cycle. Calvin-Benson cycle is a cycle, and this is not a thermodynamically infeasible cycle. So through these cycles you will have small amount of flux, just the way we do flux balance analysis. However, for this kind of reaction, if you see the minimum or the maximum flux is too much when there is no glucose, those are your potential problematic reactions which participate in these infeasible cycles. Make sense? Again, you have to turn off all the nutrient uptake, and then you have to analyze the maximum or the minimum flux that could flow through a specific reaction. Let's say you have 1000 reactions and you have identified 100 reactions that could carry huge amount of flux. Those are your potential targets. Those are the reactions that participate in cycles. That doesn't mean that those are the reactions which participate in one cycle. This 100 reactions could participate in 50 different cycles. So what is the minimum number of reaction you have to create a cycle? 3 or 2, even 2 is fine, right? If you have a bidirectional reversible reaction, even 2 is the minimum. You can create a cycle with 2 reactions. So this kind of analysis, finding the maximum or minimum flux, this is known as flux variability analysis. I was planning to talk about it later, but since we are discussing it. So this is called flux variability analysis, which is basically finding out the Vmax and Vmin for a possible reaction. So what I would like to tell you is, if you remember the flux balance model, what we did yesterday, we have biomass as our objective, right? So what we did yesterday, we max biomass, and then we have some constraint like the mass balance constraint or conservation of mass constraint and constraint on flux range, right? That's what we did yesterday. To do flux variability analysis, you will not use VBio, but you will use all possible reactions. So this is something, whoever has ideas to create a loop in any language. So basically you will pick one flux, you find out the minimum and the maximum flux possible through that reaction, and then you will repeat that process for all possible flux in your reaction model. So to find out these ranges, you need to do that. Let's say you have 1000 reaction, you need to do that for 2000 times, because you are doing minimum and maximum for each of these fluxes. So these are some of the ways you can identify reactions which participate in infeasible loop. And then there are ways you can make sure that there is no flux through this kind of reaction. So I'm saying two things here. You have a model, you suspect that you might have infeasible cycles. What I explained here is how can you find those reactions. This will not solve the problem. this will help you to identify what the problem reactions are. But there are methods which is called loopless FBA, which can make sure that even if you have this kind of a flux, there will not be any flow through this loop. So, if you perform loopless FBA, it will be 1 1 1, and then you could see we are not violating any loop based conservation as Kirchhoff's law. So, if you focus on D, we are good, right, because we are not creating any cycle from A to C, just one unit. That makes sense. It has biological relevance. Are we clear? I know that I'm giving you a lot of information, but if you are not sure about everything, I just need you to understand one thing only, that this could be an issue going forward. If you don't address this kind of issue, your model will not be predictive. If your model is not predictive, you cannot design good experiment, and you are not helping the experimental biologists. That's it. Any question? Yes. So, loopless FBA I haven't discussed yet, but loopless FBA is basically, you have a model. Loopless FBA will make sure that whenever you are maximizing for something, there is no flux through this reactions. So, loopless FBA operates on a already developed model, but as a modeler, what you need to do, you need to make sure that there should not be any reactions to begin with. So, loopless FBA is kind of tackling a situation. What I am asking, you should work so that this situation doesn't happen, right? Any question? So
in order to find the infeasible loop, we keep the nutrient uptake as 0 and then find out which is the reaction giving upper and lower bound to be infinity.
Infinity, yeah. Or if you have a range like 1000, 10,000, which one is hitting the bounds or carrying very high flux?
Those reactions have problems.
Those reactions are definitely the problematic ones, yeah. All right. So, what I would like to discuss now is, why do you think this kind of thing happened? Why you think we can end up with reactions that participate in a cycle? Can you guess, like, why these things can happen? So, the question is, why in a modeling framework, when we build the model, we can have some reactions which participate in cycles? Because we don't want to do that, but it can happen sometime. The question is why. If you remember the gene protein reaction association, all it starts with your annotation, right? You have a gene, you have to have the protein annotation, what are the proteins, and from protein you can determine what are the reactions associated with this. I know that this screen doesn't look that good, but if you look into the TV screen here, you could see that we are thinking about one conversion and one conversion only. However, in the model, for some reason, we have two different reactions. One only goes in the forward direction, the other could be reversible. Now, could you see that these two could create a cycle? So, to begin with, you should not have both the reactions, because what is the point of having both the reactions? These are the same reactions. If you afford just the reversible reaction, you are good. You don't need the irreversible one. So, it's an error from the modeling task. Sometimes we create a model automatically, as K-base does, we will see this afternoon. So, you can have duplicate reactions. You can have reactions which are doing the same thing, one irreversibly, the other one reversibly. In that process, you are creating a cycle. So, the quick fix would be get rid of the irreversible one, your problem is solved, right? That is just one case. The second case is lumped reaction. and you should have an idea. If you recall the small network that I have shown, A can produce C, or A can produce B, and B can produce C. So A to C is a lumped reaction. I lumped two steps into one, or I could have had just A to B, B to C. In that way, I have more information, I am not losing any information. It can happen in the modeling framework that you might end up with this kind of reaction, where you have the lumped reaction from citrate to isocitrate, or you can have an intermediate step. For some reason, you have both the reactions. You don't have both the reactions. You don't have to have both the reactions. If you have, and if these are reversible reaction, then you could see that you are creating a cycle. So get rid of the lumped one. Then you are good, because then you have just one step from here to here, and from one step from here to here, and through this process you are creating isocitrate from citrate. So first I showed you duplicate reaction. The second issue could be having both lumped and stepwise reaction. The third one is cofactor specificity. What do I mean by that? Again, you are basically talking about same kind of conversion. In one you have NAD, NADH, in the other you have NADP, NADPH. Most of the times in a given reaction you should not have both NAD, NADH or NADP, NADPH. It could be one or the other. In one system, one is preferred over the other. So when you are building a model automatically, sometimes you might include both, which should not happen. So in this process you are creating a cycle. So what you need to do? Ideally, you need to look into literature, you need to find out what actually happens for this reaction. If you cannot find that out, you need to find out for this kind of reaction, does this organism prefer NAD, NADH or NADP, NADPH? Based on that, you have to modify and get rid of one of the reactions, as happened here. So basically we got rid of NAD, NADH. In that case, you have just one conversion, you don't have any loop anymore. So I talked about, yeah. 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 Yeah, I understand your point. So the question was, when am I getting rid of this reaction? Am I losing information? The answer is yes, I am, because in some of the system both are present. I know that. So one of the ways you can handle it is like design a switch. So you should say not both of them should be active at the same time. It's one or the other. If the other one is active, then this one should not. You don't need to take it out. I'm just proving my point that these are the ways you can handle it. So if you define a switch, yeah, you don't have to take the entire reaction out. You can design a switch. You can create an argument, and based on that you can basically take care of this. Does that answer your question? So we talked about lumped reaction, we talked about duplicate reactions, and we talked about cofactor specificity. There are many other cases, but these are the typical cases that you will encounter when you are building a model. Any question? So what we have discussed in the last 10 minutes, I tried to convince you why mass balance is important when you are defining your reactions, and then I tried to convince you about infeasible cycles and why it could be an issue going forward designing good experiments. All right. So now when you have a metabolic model, as we have discussed yesterday, you have to define all these macromolecules. You need to make sure that they can be produced, and we need to produce cofactors or the energy molecules to drive the processes, as I have discussed. And then and only then you can simulate the model for different conditions. It could be biomass, it could be different products, as I said previously, some of the products are growth coupled, some of them are not. So, you need to know what kind of product you are targeting, whether it is growth coupled or not. So, how can you know if a product is growth coupled or not? What is the simplest way you can find out if you are targeting a product which is growth coupled or not? The clue is, think about the pipe flow diagram and think about what we do in the FBA. Yes.
Not giving that supplement to the media, the organism can't grow
Mm-hmm.
without that.
True, but what I am asking, think from a modeling perspective. What you said is right, but let us say I have a model and I would like to target a product. I would like to know if it is growth coupled or not. How do you do that? So, what will happen for a product which is growth uncoupled? If you would like to maximize that product, do you think you will get any biomass? Again, just think from a flux balance analysis perspective. You have a growth uncoupled reaction. If you maximize it, what the cell will do? In silico cell, not the actual cell. It will spend all its resources to create that product, right? Through this process, you will not get any biomass, because biomass is not coupled. That will be not the case when you are dealing with a growth coupled product. If you maximize that through this process, you will have some biomass. So, that is the simplest way you can test if you are targeting a growth coupled or uncoupled product. Okay. So, now the next topic that I would like to discuss is network gap, or gap in a metabolic network. We all know about different kinds of networks, and we know that networks are well connected. However, gaps do exist in network, right? Why do you think we have gaps in metabolic network or any network? What is the sole reason or the most prevalent reason you can think of which could cause this kind of gap? So, in this diagram, basically we are interested to produce this metabolite xanthosine, and you could see it is not connected. So, if we have this reaction here, hypothetically, we could have produced that, right? Or if you have this reaction here, we could have produced that, but that is not the case. My question is, why do you think this can happen sometimes to begin with? Why?
Hypothetical protein, the function of the protein.
Very good point. So, hypothetical proteins, because maybe there is a protein, maybe that protein does this kind of job, but it has not been characterized. Anything else? So, the broad answer to this question is issue with annotation. Hypothetical protein is one of them, unknown protein is the other one. So, we will always have these two types of proteins which are not characterized by computational predictions or by experimental measures. So, it is highly likely that the organism for which you are building this model can produce this metabolite. Someone might have already measured it, but when we build the model, we cannot create a pathway. So, you need to figure out, now we have a metabolic gap, how can we make sure that does not happen in your metabolic network? Because if you have gap, and let us say, to give you an idea, I asked you, tell me what is the maximum xanthosine that could be produced in this model. Can you do that in the given condition? If I asked you, show me the maximum xanthosine production in a given condition, can you do that? Can xanthosine even be produced here in this condition? No, because it is not connected. Like, tell me how xanthosine can be produced, because it is not even connected with XMP or xanthine. these are the ways it can produce, right? So, what you need to make sure as a modeler, you need to fill this gap. You need to fill the gap between XMP and xanthocene, or you need to fill the gap between guanine and xanthocene. If you do that, then maybe you can produce xanthocene. Again, just try to understand why having metabolic network gap could be detrimental for your model, because you cannot exploit the full potential of the model, right? So, do you all understand why sometimes this can happen? Because we have hypothetical and unknown proteins. Okay. How do you think we can fill this gap? Some of you are already doing it in the protein domain or in the gene. In the reaction domain, how can we do that? How can we fill this gap? What about orthologous genes? What about performing blast? I'm sure that definitely many of you have done it. Many of you, as we discussed yesterday, when we could not find a gene, we try to see if some of the hypothetical or unknown proteins could be characterized by performing blast against a closely related neighbor. So, hypothetically, if you can find an ortholog from a closely related neighbor, you could assume that maybe this functionality also exists in your organism, right? So, what we have found is for this organism, these steps are missing. We have definitely looked for it, but they are not there. So, when we looked into some closely related neighbors, we could see that this reaction exists, this reaction exists, and this reaction exists as well. So, now do you think your metabolic network is well connected? Can xanthocene can be produced? Can it be produced now? If I ask you, can you produce xanthocene? The answer is yes, you can, but you have more than few options. You should not like accept all of them. When you know these are the many different ways you can fix the problem, now you have to answer the question, which is the most or more plausible ones. So, what we have found that these are the two which we cannot find in closely related organisms. So, it would be highly unlikely that you do have these functionalities in them. So, if you do not have these two, that means this path will not be good, right? This is not there. So, the only way it could be fixed either this way or this way, right? Now, let us consider these steps. So, if you have this reaction here, then you are creating a cycle. Follow the dotted lines, just follow the dotted lines. If you afford this reaction, you are creating an infeasible cycle, and as a modeler we have already discussed, we should not do that. So, you cannot accept this reaction, you cannot accept this reaction, that leaves one reaction only, which is this reaction. So, if you have that reaction only, do you think your problem is fixed? Yes. So, what I am trying to get here is that when you are trying to fix a problem, you will have alternate solutions, but you should not accept all of them. You have to analyze, you have to rationalize, and then you have to accept. Yes?
So what about guanosine then? In the same cycle we have another gap.
You mean, yeah, so this is still, you think that this is still a gap? For this example, yes, but this is basically part of a bigger model. So, that is connected to some other metabolites which I did not show here. So, I was just proving my point by focusing on one metabolite, but you are absolutely right, that could still be a problem. But the question is, your problem metabolite is fixed, that gap has been filled, right? So, are we clear about why bridging network gaps is important? Is there any confusion? So, let me tell you one final statement on the network gaps. No matter what you do, you will have gaps in your model, no matter what you do. The idea is to minimize that. The idea is to reduce the number of gaps. Typically, in a metabolic network model, 20 to 30% reactions do not carry any flux, and these is mostly due to the unknown or hypothetical proteins. As long as you have some reactions which are not from primary metabolism or the metabolites of your interest, the model can still generate plausible hypotheses.
These are secondary metabolites, right?
But yeah, these are secondary metabolites, but if you are dealing with the secondary metabolites, the metabolites that you need to counter a stress or as a stress response measure, then you are in trouble. So, your model is good and it depends on what kind of problem you are dealing with. If you are dealing with all secondary metabolites, if you are dealing with a stress condition where you have to produce these metabolites which cannot be produced in your model at the moment, then your model is not good. So, you have to find the purpose, and accordingly you have to fill the gaps, accordingly you have to refine your model. I think I am kind of at the end. I think I have 2 more slides, so bear with me. So, what we have discussed, that to give you a quick recap on the metabolic model development steps. The first step is obviously to track DNA, RNA, protein and have a list of reactions. The second step is what? You have to balance, you have to get rid of the cycles, and I know that this is not clear, but if you look into that, this is how a typical genome scale metabolic network looks like. The dots are again the metabolites, the edges are the reactions, and as I said, biomass basically channels all the metabolites that we need to define growth. And finally, step 4 is to make sure that your metabolic network is well connected. Once you do that, then and only then you can define your stoichiometric matrix as we have discussed yesterday, and then you can do flux balance analysis. So, these are 4 or 5 typical steps from model building to model analysis. Any question on any of these steps? All right. I think this is one of the important slides to basically understand why flux balance analysis could identify an optimal solution given the constraint. So, just to walk you through, this is your reaction network model. Based on that, you can define the stoichiometric matrix, and when you define the stoichiometric matrix, basically you are defining a space. Here it is very easy to see because I am showing you just flux 1, flux 2 and flux 3. In real life you have 1000 reactions. I cannot possibly draw it on the screen or on a piece of paper, but you have to project this idea, and at the end we are defining this space which is called null space. Null space is the space which is basically define what is possible. So, these are the possibilities. Now, your optimal solution will be a point inside that null space, and based on the constraint you can define that. How? Again, this is the second last slide before you have a break. You can have regulatory constraint. You can tell me if you have a negative impact, or in other words you have gene repression or protein inhibition, or you have positive impact such as induction or activation of protein. If you have one of these two, that means that the flux through that specific reaction will be either turned on or turned off, or the ranges would be modified, because as I said, we operate in the reaction domain, and all this information that you have, these are either in the gene or the protein domain. It is your job to interpret that information and incorporate that in the reaction domain. If you do that, then basically this is what like this one part of the polyhedral looks like. So, I am looking at this one edge or this edge, and again this is my objective function, let us say. So, I am looking at this side, and what I am saying you can define all these different constraints, and then based on that you can get rid of so many things. So, for instance, when you define all these different constraints, these are not possible, these are out of the question. You have only these possibilities. Among all these points, you will get this, because that will guarantee that your biomass has the maximum value. Why that point? That is the answer. Why that point? Because that is the point for which your biomass is maximum. That is why it is the optimal point. But again, the point that I am trying to make here, if you define all these constraints, you are reducing the space. Otherwise, I have to look for all the points here as well, which I do not want to. If you can reduce this space, I am only operating in this space and I can quickly find my optimal solution. Otherwise, I have to look for all the different points in this space. That is the reason why, if we have more constraints, if we have more regulatory information, that will be useful for your model. So, yes. For a metabolic network model, again it is a busy slide, so let me simplify that. You operate in the metabolic level. You do not deal with transcription, translation or regulation. Whatever you can do in these two has to be incorporated in the metabolic network model as a secondary piece of information. And I think I can quickly go over this, because we have already discussed, right, how you can develop the mass balance equation for a given metabolite, and from that how can you develop the stoichiometric matrix, and this is plain and simple flux balance analysis as we have discussed yesterday. So, I will quickly go through this. So, when you have a metabolic network, few things can happen. So, this is your metabolic network. Based on the reversibility or irreversibility, you have different kind of flux ranges. If you know that information, you could see like this are the typical reaction fluxes. You have 4. To begin with, all the reactions were reversible. Now, let us say you found out that I know that 2 of the reactions are irreversible, 2 are reversible, which is why you see the range for this flux has been modified. So, it was between negative and positive. As soon as you have the delta G value, it becomes 0 to some value, 0 to some value. And then let us say you did some proteomics experiments, you know from the enzyme capacity this is the range, you can further modify it. If you have the mass balance constraint, that will also make sure that out of this range you can take just one value. And finally, you have kinetic information. So, you can measure how much glucose a system is uptaking, and that could also help you to find out the solution. So, if you go from the left to the right hand side, even if you do not understand everything, what I need you to take from this slide is how I started with a huge range and I am reducing the scope, and finally I end up with one value and one value only. The more information you have, the better it is for me to model the system. Does it make sense? Are you confused? So, we have only discussed one objective, which is biomass, but in this modeling framework, these are many different possibilities. You could minimize ATP production, maximize ATP production, you can maximize the product level of your products, you can do so many other things. Which objective you need to pick? Again, that depends. For exponential growth phase, it is biomass. For stationary phase or other phases, it could be a combination of multiple objective functions. That is a different problem. If I have time, I will discuss, but to give you an idea, these are few examples. Again, this is not an exhaustive list. There are many other possibilities which are even not here. So, to summarize, this is the last slide. We have talked about constraint based reconstruction and analysis, which is commonly known as COBRA. You will see this term in literature. Most of these are based on linear programming. However, all these other methods are sometimes used, and some of them we will discuss in the tutorial. But one thing I would like you to remember, by having all this regulatory or range, flux range information, you are defining your solution space. The more information you provide, your solution space will be reduced. Finding an optimal value would be easy. So with that, I think let's take a 5 minutes break.
Yeah.
And the next lecture will focus on photosynthetic microbe model development. But before we leave, is there any question you have? I know that you mentioned about transport and exchange reaction, which I'll explain in the next class. But before you take the break, is there any question that you want to ask? If not, let's take the break. Steps for a photosynthetic microbe, I would like to address two issues. So first of all, I was asked about the transport or the exchange reaction. Ask your question again so that the class can understand. So what is the issue with the transport or the exchange reaction? Is it why we need them, or we know we need them but how to define them? Which is? The second one. The second one. Okay, I got it. So just to recap or refresh your memory, by transport reaction basically I mean, let's say A can produce B, B can produce C, and C is your final product or the product of your interest, and you want to make sure that C is coming out of the cell, C is being excreted from the cell. For that, you have to define a transport reaction, or in other words, you have to have a transporter which can take C out of the cell. If you don't have a transporter in your given system, you cannot hypothetically just add a transporter. That will not do any good. From the experimental point of view, you need to make sure that you can design that transport process. But let's say hypothetically you have a transporter. So for that, your reaction will be C to CT, meaning that C what is transported, and then from the transported C, it will be excreted in the outside environment from CT to CE, which is the final product. Now this is how you can have a transport and an exchange reaction. The only difference is, in modeling framework you cannot have a CE here, because what will happen if you have a CE, then CE only participates in one reaction. Whichever metabolite participates in one reaction, that's a problem metabolite. That will force, there is no reaction through that, there is no flux through that reaction. So the final reaction for your exchange, it would be CT just going out. So in other words, your exchange reaction would be CT going out. On the right hand side, you don't have to define anything. That's how you will define an exchange reaction. Does it make sense? Can you see what will happen when you are taking a nutrient? So you are taking a nutrient inside the cell. It's the opposite. So from here your glucose is coming near your cell. So here you will define glucose transported like this. On the left side you have nothing, and then glucose transported will be inside, which is glucose transported, glucose, let's say in cytosol, then you are good. So depending on if you are excreting a metabolite or you are uptaking a metabolite, your exchange and transport reaction would be different. Does it make sense? One other question that I got about different time points and switching of different regulations. Remember, flux balance analysis would let you solve the model or simulate the model for a specific time point. It's not a dynamic analysis. You are not doing it for the entire cell cycle. You can only do it for a given time point. So it is of utmost importance to define that time point. If you, let's say this is time point 1 and this is time point 2. So, time point 1 is the mid exponential growth phase, time point 2 is at the beginning of the stationary phase or the end of the exponential growth phase. As a modeler, I need to know what conditions that are different. Conditions could be of different type. It could be, well, we know that these genes express more, these proteins have higher abundance, or we know that these are the switches that operate in here versus here. As long as you give me this information, I can simulate the model, and then and only then the model will generate different kind of results. But let us assume that between point 1 and 2, you did not give me any useful information. You did not tell me how these 2 points are different. Then if you simulate the model, you will end up with the similar kind of results. To get good results, to get good predictions, you have to have good conditions. If you do not have, then you will not, you cannot simulate the model well. You cannot represent the conditions well. Does it make sense? So, make sure you are absolutely crystal clear about the scope of flux balance analysis. It is for a specific time point. It is not for the entire cell cycle. Does it make sense? Any other question? All right. If not, so let us look at photosynthetic microbes. So, basically some of the things I have already discussed yesterday. So, though for those slides I will go faster, but stop me if you need to discuss about something. So, I will first discuss about the electron transport chain, then I will briefly discuss about the carbon fixation mechanism. I will talk about these things specifically, how you can use the model to make sure a specific carbohydrate could be stored, because if you remember, it is a steady state analysis. We really do not allow something to store in the cell, but there is a way, there is a workaround. So, I will explain how that could be done. And finally, I am sure many of you have encountered this issue where you are dealing with a polymer with a value of n, and n could take between 100 and 400, let us say. In the modeling framework, how can you deal with that? I will explain that. And finally, I will touch on some of the things that Amit is expert of. So, 13 CMFA and how that could be used. So, I will briefly mention it because you have a specific class for that. So, I do not want to take any time explaining that, because that will be explained anyways, and how can you use existing data which is useful for your model. All right. So, again, this is a busy diagram. What I need you to look at is basically different kinds of bacteria. So, if you look on the left hand side, it is one form of bacteria, it is called acidobacteria, cyanobacteria, anoxygenic bacteria, but this is filamentous, and purple bacteria. I got this question about electron transport chains from some of you. So, what I need you to understand is, look at this. What are the donors for all these 4 cases, and how electrons are being transferred? Again, you will see there are multiple different compounds here. Quinones and cytochromes, they are the known electron carriers. You will also see a common theme of cyclic electron flow. For this case, purple bacteria, you could see electron comes from either organics, sulfide, or hydrogen. For instance, the bug that I work with, it is a purple non-sulfur bacterium. So, it takes its electron from an organic source or an inorganic source, depending on which mode I am operating. If you remember, most of these bugs, they are pretty flexible. They can photosynthesize, they can even not photosynthesize. If you do not have light, they can still survive if you provide a different carbon source. For instance, my bug can survive with succinate. If you do not provide any light, it is fine, it can still survive. In cyanobacteria, they are also very flexible. they can survive with carbon dioxide and light, they can survive with just plain and simple sugar as E. coli. They can also survive with sugar plus light if you do not provide carbon dioxide, mixotrophic growth. So, what I am basically telling you is that these organisms are flexible, they can switch from one mode to another depending on what is out there, what are you giving it. Although it is possible in one mode of operation is more efficient than the other, that is possible. But what I need you to take from this slide is basically, metabolic processes are pretty flexible in these microbes. So, that is the take home message here. Even if you do not want to go to the details of this electron transport, that is the message that I am giving you here. I know that two of you have asked me about electron transport chain. Do you have any specific question at the moment? All right. Anyone else? All right. Okay. Now, for a non-photosynthetic microbe, if it is a unicellular organism, you will know that in unicellular organism we have an outer membrane, inner membrane and cytosol, that is it. Prokaryotes does not have any cell wall. Yes, I saw a hand, please. Flexible means, okay. Flexible means it could basically survive in multiple different environment with multiple different carbon sources. So, if you think about a non-photosynthetic microbe, a non-photosynthetic microbe can survive with a few different carbon sources. You will not see this kind of flexibility in a non-photosynthetic microbe. So, for instance, I know that you folks work with yeast, right? What is the carbon source when you grow yeast in your lab? Dextrose. So, there are like commercially available medium, there are only few of them, but more or less the major carbon source is same, sometimes the nutrients are different, more or less something like this. You will not find 3 or 4 different modes of operation for a non-photosynthetic microbe, because they do not have this kind of flexibility in their metabolism. They do not have a photosynthetic electron transport chain, right. So, that is what I am saying. So,
either glucose or galactose.
Right. Some of the organisms, they can take C5 or C6 sugars, but sugars, that is all. For a photosynthetic microbe, you saw that they are pretty flexible. All right. So, for a non-photosynthetic microbe, as I was saying, you will have outer membrane, inner membrane, but you will not have cell wall. Prokaryotes do not have cell wall, and you have cytosol, and that is what you will model. So, you will model the cytoplasm and extracellular space, that is all. You will have 2 compartments. When you are dealing with a photosynthetic microbe, the space between the inner and outer membrane, this is known as periplasm. This space is pretty interesting for a photosynthetic microbe. Look at the surface. It is not only here, it goes there. So, it is pretty significant. Unlike this, a non-photosynthetic microbe, a photosynthetic microbe, you cannot exclude periplasm. Periplasm is significant. Why it is significant? The electron transport chain that I mentioned, it is in here for a photosynthetic microbe. So, you cannot exclude that, or in other words, you will have 3 compartments. You will have cytosol, you will have extracellular space, and you have periplasm. For cyanobacteria specifically, as I told you yesterday, it has a different intracellular or a subcellular compartment which is known as carboxysm. Carboxysm is known to concentrate carbon dioxide to bicarbonate, and then it uses the bicarbonate in the Calvin-Benson cycle process. So,
is there any metabolite in periplasm?
Other than the electron transport chain, no, but it is involved in the transport of different metabolites. So, basically, unlike E. coli, you will have to define like for A going in, so it will come to the periplasm first, from periplasm, then it will go to the cytosol. So, you have to define just one more reaction, but it is nothing. So, in that way you can have that extra compartment.
Give a transporter protein definition.
Yeah, yes. Any question? Okay, you might have seen this figure many times. This is a typical electron transport chain that you will find for a cyanobacteria, in a cyanobacteria or in a plant species. So, this is photosystem 2 and photosystem 1, and in between there are multiple other compounds. So, in other words, water gets oxidized, gives away the electron moves through this plastoquinone to the cytochrome, it goes to photosystem 1, and then it moves to ferredoxin, and by using this electron you can basically produce NADPH from NADP, and this water breakdown product proton, they will be pumped through ATPase, and in the ATPase or ATP synthase basically you will produce ATP. So, this process will have a linear electron transport and also a cyclic electron transport. By doing this, what you are doing, you are producing the energy that you need to power your metabolism. You are creating ATP, you are creating NADPH. But again, remember this is what you will find in a cyanobacteria, in a cyanobacterial species or plant. For others, it will be different, but the concept is still the same. Electron transport chain is involved to produce energy. One thing that I would like to highlight here is what you will see this afternoon, that we will use a tool to develop a model automatically. Many times it happens that, for instance, let us say I am working with a purple non-sulfur bacteria. I know that the electron transport chain is not like that. It is possible that when I develop a model automatically, this is what I will get, which is not right. So, if I were you and if I am developing a metabolic model through an automated tool, as soon as I have the list of reactions, I would go and I will see what kind of electron transport chain is there, the first check. What kind of carbon fixation is there, second check. If these two are not right, you have to manually fix them, because remember, those automated models are just draft models. You cannot trust them, the quality will not be that high. So, you need to do this manual refinement. One other thing, it might sound weird, in a modeling framework we consider photon as a metabolite, because if you do not consider photon as a metabolite, you cannot model it. We need to model it, because as I said, cyanobacteria, when it operates in phototropic conditions, meaning light and carbon dioxide, light is the limiting nutrient, not carbon dioxide in most of the time. So, if you do not have light in the model, how can you define that? If you look at the reaction that happens here, so water breaks down and produces proton and oxygen, right? It does not show any photon here. In real life, when you will build a model, you have to define photon like this. So, you will have water and you will define photon like this, literally like this. It might sound weird, it might look weird, but this is what it is. You have to have photon as a reactant in this reaction, and then on the right side you will say water and oxygen. Does it make sense? Even if it does not, for modeling purpose, this is what you have to do. What else? Yeah. So, we will assume that light is being used in both these photosystems, because cyanobacteria have two different photosystems. Purple non-sulfur bacterium has one photosystem, as we have discussed yesterday. So, depending on what kind of organism, what kind of photosynthetic organism you are building the model for, you have to define the this kind of reactions basically. And again, when you are defining the photon, depending on which photosystem you are operating, you can define the wavelength. For instance, light is light, it has all this different spectrum, right? So, in the visible spectrum we know the range, but we also know for photosystems 2 the wavelength that it operates is 680, for photosystem 1 it is 700. So, you can even define two different photon, one as 680 the other one as 700. You can define this reaction like this. So, in that way you are not mixing two different light sorts of light wavelengths. So, that will make sure that for this photosystem you are operating in one spectrum, one part, and this one is being operated in a different spectrum. So, in that way, so what I am basically telling you, to make sure that you are modeling the organism in the proper way, so that you can perform flux balance analysis, you have to do all these minor tweaks, because otherwise you will not be able to, like, simulate the model well. Some of the times it looks comical that we are thinking photon as a metabolite, but it is what it is. Any question? Yeah, so I have already mentioned it. So, depending on what kind of organism, you need to figure out what are the electron transport chains. Based on that, you have to model it accordingly. All right. So, again, you have seen these slides yesterday. You have seen that depending on if it's an oxygenic or anoxygenic, your right hand side of this reaction will be different. The difference between these two is who is providing the electron, what are the electron donors. Depending on that, your products on the right hand side will be different. And we also have discussed the Z scheme, which I will not repeat myself. One thing that I would like to do, because I was asked that question, you asked that question, like, what do you mean by flexibility? So, this is what I mean by flexibility. There are organisms which can operate in many different modes. In the phototrophic mode, it takes photon, it takes carbon dioxide or maybe H2S to produce this. So, likewise you can go through these maps and you can see how flexible this organism could be. I'm not saying all of them could do all 4 of them, but many of these cyanos, many of these purple bacterium, they operate in many different modes. Depending on what mode they operate, the electron donors could be different. It could be inorganic or organic. For instance, again I am giving the example from my bug. So, in anaerobic condition, it could operate on lignin breakdown product, but it will require light. In aerobic condition, it could survive with organic carbon sources, and it does not require light. So, just to give you an example how flexible these photosynthetic microbes could be. So, it depends which mode is better suitable for you. How do you decide? Can you think about a way, or why do you think we have to operate a photosynthetic microbe in one mode versus other? What would be the deciding factor? So, yeah, we understand these organisms are flexible. So what? That's the question I am asking. In which mode the biomass concentration is high, so the product is most. That's one answer. Anything else?
The product which I am required from our study,
yeah.
The product of my interest. Which condition the microorganism will produce the product of my interest?
Excellent. That's another answer. Anything else? Based on the geographic location, it's possible that one of these modes will be preferable than the others. Okay. Anything else?
Economic source of economy.
Source of economy. Let's say one of these carbon sources is cheaper than the other. Anything else? Good. I think you pretty much all 4 of you have covered the bases. So, yes, first of all, which mode, that will depend on what is our objective, what actually we want to do here. If you remember, yesterday I talked about backward design. So, first of all, we have to define our problem. So, again, a very crude example, let us say we would like to produce a product and we know this microorganism can do that. The first question we should be asking ourselves, can it be done under photosynthetic mode or non-photosynthetic mode? Okay, let us say under photosynthetic mode. Well, in that case, what are the other conditions? What are the other nutrients it needs? You have to define that, and then you have to see whether we could reach to the desired level. Most of the times we cannot, and then we have to think about designing or engineering that bug. That's how you approach. Flexibility is good, but at the end of the day, we need to figure how the flexibility is helping us, because otherwise, yes, from the point of view of science, it's good, but these days you need to have at least an engineering aspect. You need to have an application aspect, because everyone will ask you that question. So what? Yes, this is good, this is exciting, but so what? How can this be useful? And like everywhere it's the same. And I was talking to my colleagues here, it's even more true in India. If you cannot show in a project how something could be used for an application, it's very difficult to get funded. So that's something that you have to remember all the time. All right. Okay. Again, I'll quickly go through it just to show you. Yesterday we talked about 3 different mechanisms. I'm showing 2 here, but we talked about Calvin-Benson cycle and hydroxypropionate cycle, and we have discussed that different microbes, they use different ways to capture carbon dioxide. It's not one way. It could be one of the 3 ways. I'm showing 2 here. Calvin-Benson cycle is the most prevalent one because it's used by cyanobacteria, it's used by plant. This is something that some purple bacteria used. So one common theme across these 2 is both of them are energy intensive process. This one is more than this because it requires 9 ATP and 5 NADPH, whereas this one will require 5 NADPH and 5 ATP. So again, if I were a modeler, if I am building a metabolic network model and I am using an automated tool, as I said, first thing I will check, what about the electron transport chain? Is it the same that I know? As long as you know what kind of microbe for which you want to build the model for, you can go back in literature and see what kind of electron transport chain should be there. Similarly, you should also know what kind of carbon fixation mechanism should be there. As long as these 2 are right, then you can proceed. Most of the times they will not be right, and we will see in the tutorial this afternoon. Then you have to go and you have to fix them. But these 2, I'm probably repeating myself. These are the 2 most important things when you are doing or when you are building a metabolic model for a photosynthetic microbe. Without doing these 2 right, there is no point of building a metabolic network model for a photosynthetic microbe. Any question? Again, cycle fixation is important, and I have already given you this example. Is there any question on that? So what I am basically doing here is that if you remember in the first lecture, I already discussed why balancing a reaction is important. I said why not having infeasible cycle is important. These are typical requirements no matter what kind of models you build. On top of these 2, you have these 2 requirements when you build a photosynthetic model. Does it make sense? So all these requirements, they are related to what kind of biological system you are dealing with. Some of the biological systems, they have specific characteristics. You need to make sure that you capture that characteristics when you are building the model. All right. How many of you have heard about diurnal cycle or circadian cycle? Okay. What is the circadian cycle? So anyone? Go ahead.
The total cycle is composed of 24 hours and maybe equal 12 plus 12, otherwise.
So in other words, it's related to the rotation of the earth, and we know like how long it takes, 24 hours, and then as you mentioned, like with 12 it could be little more or less, but it's related to the rotation of the earth and how some of the things could get repeated with this process. What about diurnal cycle? That is kind of tricky. So, does anyone know what's the difference between diurnal and circadian cycle? So, diurnal cycle is dependent on the presence or absence of light. So, like unlike circadian rhythm, which will always happen because the earth is rotating and it's not stopping anytime soon, I hope. So, circadian rhythm is circadian rhythm, and many of our processes, like by us means all the living beings, many of the processes are being controlled or regulated by this rhythm. But for cyano, it's especially interesting because they have some of the processes which are regulated by circadian rhythm, but they have many processes which are regulated by the availability of light, presence or absence of light. If you shift this availability, let us say you have done an experiment 12-12, you have done an experiment 18 hours of light, 6 hours of dark, and like someone else has done the other way around, you will see the differences. Maybe someone has done the experiment with continuous light for 24 hours, you will see the differences. So, capturing the diurnal rhythm in a modeling framework is of utmost importance when you are building a photosynthetic microbial model. Why? Because many of the photosynthetic microbes, they produce a carbon storage. They use the light in the light phase, they store the carbon in glycogen, sometimes in sucrose, sometimes in other carbohydrate form. And if you remember one diagram I showed yesterday, at night time it uses the carbon as a this carbon storage as a carbon source, and then it powers its metabolism. So, you need to make sure that you can afford the storage molecule in your modeling framework. If you could remember, I told you that that could be a problem, because we operate with pseudo steady state, meaning that no accumulation, nothing could be accumulated. The question is, how can we get around with this? There should be a way to get around with, because if we don't, then what's the point of doing this modeling exercise? Do all of us understand the problem? Okay. Now, I will limit myself to one example, which is glycogen, and that's something cyanobacteria could store in the light and uses in the dark. So, let us assume that's the flux value 0.99, which basically needs to be stored. If that's the case, how do you do that? How do you model an internal sink or an internal source? So, in the light phase, light uptake is obviously greater than 0, and you are creating a demand reaction. How can you create a demand reaction in a metabolic network model? You have to create a demand and a sink reaction. Remember, at the beginning of this class, I gave you the clues. I gave you how can you define a demand and a sink reaction when we just discussed about excreting a metabolite and bringing a metabolite in. It's exactly same. Can anyone give me the answer? How we can model it?
For sink reaction, the RHS will be 0.
Yeah.
Just keep it as a defined thing. For demand one, the LH.
Absolutely. That's what it is. So, this is how. So, when you are defining the storage, your cytosolic glycogen will be on the left hand side, the right hand side you do not need anything, because that is a storage. If you have something there, let us say hypothetically, you have cytosolic glycogen as a reactant, and let us say A as a product, but since A will not participate in any other reaction, this reaction will not carry any flux. So, you should not do that.
Just like a biomass reaction, right?
Just like a biomass equation, as we have discussed yesterday, yes. And same thing for the sink reaction, same thing for the demand reaction. So, basically you will have on the right hand side, you will have cytosolic glycogen, sorry, I said the other way around, but you got the point. So, demand reaction is like this, storage reaction is like this, because you are storing glycogen, and that's how you can afford a sink and a demand in the model. So, what will happen in the light phase? You will have the storage. So, this reaction can carry flux. You know what's the flux value. At the night time, you will make sure that this reaction has a maximum flux value which is equal to that. Does it make sense? So, yesterday we will talk a lot about this, but what I am telling you, if you could look into this diagram, so it basically shows how the gene expression changes, and this is one example in the dark light phase. You could see from this, and again I do not expect you to read these, but what I am basically showing here, for some of the reactions it will be peaked in the light, and so it will be upregulated in the light phase and downregulated in the dark phase and other way around. There are examples. So, we will discuss about them in great detail tomorrow, but what I am basically trying to show here is, based on the diurnal rhythm, there will be genes which will behave differently. Any question? Okay. I think I have two other things. I will try to wrap it up in the next 5 minutes. So, as I told you, we have to deal with polymers when we build a metabolic network model. So, polymeric material like ubiquinone, which is an important electron transporter, or peptidoglycan, which involved in the cell wall, again very important, we cannot exclude them. How to deal with that? Because in the modeling framework, if you just define N, you will not get any flux, because in our modeling framework we have to define the stoichiometry. N is just generic. You need to tell me what's the value of N. So, how to deal with that? I think the answer is on the screen, but still if you could think about it. How can we deal with the polymers in a metabolic network? So, what basically happens here? Let us say you have a monomer, right? To make the polymer, you have to just have the repeat units, maybe 10 times, 100 times, 400 times. As long as you can tell me that this polymer is made of this many units, and through this process we have to spend this much of energy. By energy I means in the terms of NADH, ATP or NADPH. If you do not have that information, then restrict yourself just to the monomer. So, as long as the model can produce 1 unit of monomer, you are fine. Or if you know the value of N, define that N. One of the two ways, either you define the monomer and just end the reaction there without showing the polymeric reaction, or you show the polymeric reaction by defining N. But if you do that, if you show the polymeric reaction, you need to show if that reaction would need energy. You have to fully define that reaction, otherwise it does not make sense. So, that is how you can tackle this issue in a metabolic network. Any question? So, just to quickly recap what we just discussed. First thing, how can you deal with a sink or a demand reaction, and it is of utmost importance because we are dealing with a photosynthetic microbe which stores carbon in the light phase and uses carbon in the dark phase. So, in the metabolic network model you have to afford that. Second, we have discussed many times we have to deal with polymeric compound or polymeric metabolites. The question is, how can you model it? What I told, either you end the reaction with one monomer, A plus B making one monomer, done, or if you know how many subunits you need to assemble and you know the cost for that reaction, you define. As long as you define that reaction, you can have it in the model, you are good. but you cannot have any reaction with just N by not defining N. Does it make sense? Okay. So, one of the other ways, why we are discussing all this, one of the other ways, like how can you gather more information from literature, doing some experiments to inform the model. As we have discussed, if you remember that solution space which we restricted, as long as we can do that, our life would be easy. FBA doesn't have to find or test so many different points, and we can get the solution. So, one of the ways people do that is people can label a carbon in a let's say you can label one carbon in a 6 carbon molecule glucose, and then you can see, you can basically track how the carbon basically exchange or transferred through the process, and then you measure some of the metabolites. Based on that, you can estimate flux. That is metabolic flux analysis. The issue is, for photosynthetic microbe, you are dealing with carbon dioxide, one carbon. So, that is the issue, and I think, will you discuss with So, that is what Amit will discuss in his lecture. So, I am not going to discuss at all on that, but I am just telling you the information so that you know what is the issue. For glucose, it is okay, because you have 6 carbon, you are labeling one of them, and then you are tracking how it gets exchanged to different other molecule. For carbon dioxide, it has one carbon, how to deal with that. But there is a way to deal with that, which will be the focus of that class. But hypothetically, let us assume by doing something, you have measured, you have estimated the flux. Once you have estimated the flux, that, yeah.
Sir, but for most of the mixotrophic mode of cultivation, glucose is also there, and we are also giving carbon dioxide. Then both the carbon sources are there.
Right. So, the question is, can we not do that in the mixotrophic condition? Do you want to answer that? So, yes, in the mixotrophic condition, if you have more than one, you can do that. But when I say CO2 and the issue, I mainly mean phototrophic condition. If you have just carbon dioxide.
Because in most of the cases, we see that means providing another carbon source and also providing carbon dioxide, the biomass consumption.
Yes, absolutely, absolutely.
So, why not in that mode? Means
Yeah,
most of the
no, exactly. So, the issue is, why not in that mode is, at the end of the day, our selling point is we are using cheap or cheapest possible nutrient, carbon dioxide. Now, if you are spending glucose, and at the end of the day, we have to do economic analysis to see whether that is a viable process. If that is, then why not? Yes, I agree. So, it depends on, like, what is our need and whether we can fulfill the need. But yeah.
One more thing, it will be computationally expensive. When you have 2 carbon sources, then solving those problems will be tricky.
Right.
Not difficult, but it's tricky.
So, it's dealing with the steady state and whether we can reach the steady state. Again, I am not going to spend time because he will give a lecture on it. So let us say hypothetically, you can estimate the fluxes in a metabolic network by doing this. But the issue here is, most of the times you can measure the fluxes in the central carbon metabolism or maybe in the amino acids. So, in other words, you have a model of 1000 reactions. By doing this experiment, you can measure 100 flux, let us assume, but that is not all the flux. If you have that flux estimation, you can use that to limit the flux range. So, let us say, again giving you a very crude example, you have a 100 reaction model. By doing this exercise, you have measured 10 reactions. So, basically you will define V1 up to V10, and then you will define their range. You will define their range, again you are restricting the solution space. Based on that, you can estimate what would be your other fluxes. So, that is how 13C MFA could be useful in a modeling framework. Does it make sense? Like, obviously, when I say you can estimate flux, the question should be, why we do even care? That is why we do care, because it is helping our modeling prediction. It will help our modeling prediction. Without 13 MFA we can still do that, but probably the predictions would be better or improved if we have this kind of information. In addition to that, sometimes in literature you will have growth information. You will know the doubling time based on, you can calculate the growth rate, and then you can compare your in silico growth rate with your in vivo growth, with your experimental growth rate, and then you can see how good or bad the model is. The rule of thumb is, as long as your in silico growth is in the same order of magnitude, it may not be same, it may not be very close, but in the same order of magnitude, it's okay, because the model predictions, as I said, they are qualitative predictions. You cannot take the scales like you can increase the production level by 10 times as it is. What you need to take is, yes, by doing this maybe you can increase the production level. That is the information that you should take. All right. So, many other times, what are the other information that you can use? This is the last slide. So, some of the times you can get some behavioral information. So, you might know what is the photosynthetic rate. What do I mean by that? Let us say by photosynthetic rate, basically what is the rate of the reaction of the first reaction in the electron transport chain. Sometimes you have this information. So, you can go back in the model and you can ask a reverse question. So, you should ask, I know that V10 has a flux value of this. Now, what could be the value of the nutrient uptake? That is something that you can test in the model. So, to get V10 equals to 10, what are the nutrients level you need? By nutrient level I mean carbon dioxide or light. Some of the times if you are dealing with a nitrogen fixing microbe, you can also get this kind of diagram which basically shows what is the nitrogen fixation rate, and you can also ask this kind of a question. So, what I am trying to show here is that when you are building a model, you need to really look into literature and see what kind of information is out there and how can you make the best use of them. When you are dealing with a photosynthetic microbe, this kind of information will be of utmost importance, which you do not need if you are dealing with a non-photosynthetic system. These are the additional steps or additional pain you have to go through. So, just to recap, we talked about different photosynthetic electron transport mechanism. We know that different photosynthetic microbes have different mechanisms. So, when you are building a model, you better look into that. We also have discussed multiple carbon fixation mechanism. Same thing goes for this as well. And then we have talked about the diurnal variation. We have talked about how we can use the model to create a source or a sink reaction. And finally, we have talked about how we can deal with the polymers in a metabolic modeling framework, as the metabolic model framework cannot deal with unspecified stoichiometry. You have to define your N. And finally, we also have talked about how we can use the information that is out there, let us say photosynthetic rate, nitrogen fixation rate, and how can you make use of these pieces of information to find out what are the nutrients you need so that you can create or so that you can fix that much of nitrogen. I think, yeah, that is all. Thank you. So, we will meet when? At